Good day, everyone. I'm Carlos Saleo from Great Job St. Agnes, and I'm here to present with Eleanor Pomisaran, Stephanie Almoglanque, and David Emmanuel Pereira to present our research entitled The Level of Knowledge of the Great Job St. Agnes Students in St. Mary's College of Baliwag about CRISPR and genome editing. To carry on with our chapter 1, let's move on to Miss Stephanie Almoglanque.
For starters, these chapters serve as the introduction of the research and the guide of the researchers. Its contents are mainly the significance of the study, specific question, the general problem, and the genome editing is a branch of biomedical field wherein a gene of an organism is modified. It is a method that the scientists use to change the DNA of many organisms, including plants, bacteria, and animals. CRISPR is an abbreviation for Clustered Regularly Interspaced Short Palindromic Repeats, a genome editing tool that's also known as gene editing. CRISPR can be considered a huge leap for the healthcare system. It has the potential to cure illnesses that currently have no cure. It can also improve crop engineering and produce biofuel more efficiently. Statement of the problem. For the general problem, this research aims to perceive the level of knowledge of the grade 12 St. Agnes of St. Mary's College of Baliuag about CRISPR and genome editing. This study will specifically cover the following research questions. First, what is the level of knowledge of the grade 12 students at St. Agnes about genome editing and CRISPR? Second, is there a significant difference between male and female students prior to their level of knowledge about genome editing, CRISPR, and their significance? Third, what is the significance of knowing the level of knowledge of the grade 12 St. Agnes students about genome editing and CRISPR? Fourth, how will the grade 12 St. Agnes students describe genome editing and CRISPR? Fifth, should genome editing and CRISPR be introduced in the school curriculum? Moving on to the significance of the study, this study aims to determine the level of knowledge of the grade 12 St. Agnes students about CRISPR. The main beneficiary will be the STEM students in grade 12 at St. Agnes in St. Mary's College of Baliwag. It is also appropriate for biomedical students, professors, in an institution. Finally, to the future researchers. This study should be used as a guide to create a larger study on CRISPR. For discovering the limitation, it took 3 months for the researchers to come up with the findings for the research. The main research instrument that was used is the online survey questionnaire. This study does not cover extraneous variables that may affect the level of knowledge of the grade 12 St. Agnes student.
I am Maria Eleanor Pamisaran and I will be discussing Chapter 2 of this research. This chapter contains the review of related literature, theoretical framework, conceptual framework, hypotheses of the study, and the definition of terms. Genome editing is the ability to make very exact alterations in a living organism's DNA sequence. Thus, it can alter its genetic composition. It has its own advantages and disadvantages. According to Nelia, gene editing technology can potentially speed up the drug discovery process, which can help develop cure for cancer and other diseases. If in the wrong hands, it can also inflict massive damage to the world and humanity itself. In China, a Chinese researcher, Jian Qiu, was sentenced by the Chinese local court to 3 years of imprisonment. He tampered with the genes of two embryos, which is illegal and was found out. In the Philippines, genome editing is not a new topic. In 1987, scientists from the IRRI and UPLB, the director for crops of the PCARRD, and the quarantine officer of the BPI established a group to develop biosafety protocols and guidelines for genetic engineering and activities related to research for UPLB and IRRI. In other countries, genome editing is also known. For example, in America, a gene editing experiment made some people with Leber congenital amaurosis or LCA, a form of severe vision impairment, see colors again. Genome editing does not only benefit the field of biomedical engineering, it can also help the field of agriculture. Carvolius et al. 2021 stated multiple ways on how scientists can edit crops and livestock to be resistant to climate change. such as increasing the tolerance to salinity and drought, increasing yields, making livestock more resistant to disease, increasing their nutrition, and so much more. According to Bryant, a student of the Green School in Rhode Island, he stated that gene editing is only the beginning of a safer and disease-free future. Meanwhile, Kent argued that the future of gene editing can cause humanity to lose what makes them human. Genome editing should be introduced to grade 12 students because it has so much potential. As Fernandez stated, genome editing has the potential to cure cancer, blindness, AIDS, blood disorders, and also the terrifying COVID-19. CRISPR is a revolutionary genome editing technique that allows scientists to easily change DNA sequences and gene function. It also has its own advantages and disadvantages, in which the CRISPR-Cas9 system is extremely precise in theory, but not in practice. It can cause off-target alterations, which are changes that occur elsewhere in the genome. The theoretical framework of the researchers show the current and prospective applications of genome editing and CRISPR-Cas9. For the group's conceptual framework, it shows the aim of the study, which is to know the level of knowledge of the grade 12 St. Agnes students in St. Mary's College of Baliwag about CRISPR and genome editing. For the hypothesis of the study, the null hypothesis is the grade 12 students of St. Agnes have no significant knowledge of genome editing and CRISPR. And for the alternative hypothesis is the grade 12 students of St. Agnes has significant knowledge of genome editing and CRISPR. For the definition of terms, it will be shown on the following slides.
Hi, I'm David Pereira, here to discuss the third chapter. This chapter comprises the research methodologies. For the research procedure, the researchers used a mixed method in this study. They conducted a survey research design to determine the level of knowledge of grade 12 St. Agnes students in St. Mary's College of Baliwag about CRISPR and genome editing. To address the research questions, a mixed method research design incorporates the methodologies of quantitative and qualitative research. Moving on to the respondents and sampling procedures, the researchers conveyed the respondents of the study who are the grade 12 St. Agnes students from the senior high school of St. Mary's College of Baliwag. The researchers did not use any kind of method to obtain their intended sample size from the total of 33 students because they had a predetermined number of responders, which was 33. However, instead of the original 33 respondents, who have been lost due to panel non-response, only 29 respondents from the section were able to answer. The researchers used a non-probability sampling method, particularly convenience sampling. Moving on to the research instrument utilized in the study, the researchers utilized Google Forms and Facebook Messenger to administer an online questionnaire. The questionnaire used the Likert scale, with responses ranging from 1 to 5, with 1 denoting strongly disagree and 5 denoting strongly agree. The researchers designed the questionnaire to have only 14 items. Next is a research procedure. The questionnaires were given to 29 students in grade 12 at St. Agnes of St. Mary's College in Baliwag. The researchers sent the administration of St. Mary's College of Baliwag a letter of consent to distribute the questionnaires to acquire the relevant information from the students and perform the survey. The participants were informed that the study would be kept confidential and private to avoid any problems during the process. In addition, the researchers conducted an online survey questionnaire with a professional human biologist to gain his perspective on the subject. Lastly, for the statistical treatment, the formulas and statistical tools listed below are the formulas and statistical tools used by the researchers to determine and analyze the data collected through their closed-ended survey questionnaire. Percentage. This formula is used to determine the respondents' frequency, counts, and percentage distribution of relevant variables. Weighted average. The mean is used to determine the average rating of the software evaluation. General weighted average. The average value of the data is calculated using the weighted mean. T-test. The t-test is an inferential statistic that is used to determine whether two variables are significantly different.
Moving on to chapter 4 to be presented by Mr. Carlos Vincent C. Aleo. This chapter contains the analysis and interpretation of data and information that was acquired prior to the general and specific problems as stated in the statement of the problem. The said data was acquired through the means of survey questionnaires via Messenger and Google Forms. Here is Table 4, which shows the frequency and percentage distribution of respondents according to gender. Table 5, the data of students' level of knowledge about genome editing. Table 6, the data of students' level of knowledge about CRISPR. Table 7, the data of students' level of knowledge about the significance of genome editing and CRISPR. Table 8, the mean and standard deviation interpretation for respondents' level of knowledge about genome editing, CRISPR, and their significance. Independent sample t-tests results on the level of knowledge about genome editing, CRISPR, and their significance between the male and female students. And lastly, our qualitative questionnaire for Mr. Maurice Olivier Bailan, who specializes in human biology. Moving on to Chapter 5. This is wherein it comprises the summary of findings and conclusions that were acquired from the obtained results from the respondents. In the summary of findings, the researchers aims to answer the following research questions. The first specific question concluded that the Great Job St. Agnes students are slightly knowledgeable about CRISPR and genome editing. In the second research questions, the researchers found out that there is no significant difference in the level of knowledge about genome editing, CRISPR, and their significance between male and female students. The third specific questions of the researchers concluded that genome editing and CRISPR should be introduced to the students as it might spark the students' interest in pursuing the fields of genomics, and it will be beneficial to have a lot of geneticists working in the future. The fourth specific questions based on the data acquired is concluded as the students only have slight knowledge about genome editing and CRISPR, thus they only have slight knowledge about its significance. The last specific questions, the researchers concluded that the CRISPR and genome editing should be introduced in high school and college because it is a promising innovation of science, and science itself is a fundamental part of society. Overall, the researchers concluded that the level of knowledge of the Great Job St. Agnes students are slightly knowledgeable about gene editing, CRISPR, and the significance between the two. The following slides are the recommendation of the study. That is all. Thank you.